All right. So just some announcements to start. Next week, Tuesday, is going to be our first test. If you looked at the slides I posted for today, I just posted them this morning. If you go down to slide like 25 or something like that, you'll see there that there's a slide that says past this point, information won't be on test one. So basically the end of test one material will be our glycogen breakdown and our glycogen synthesis. So that's what the cutoff is going to be. So for next week, the test is going to be, you have 24 hours to take it. So it's going to be open 12 a.m. on the 16th and close at 11:59 p.m. Since I'm doing Tuesdays and Thursdays for the lecture, I'm going to do the same thing for the test, in that you have 75 minutes to take it. It's going to be similar to the tests I gave in biochemistry. So look for a good proportion of short answer questions. I will also throw in multiple choice questions for that. As of right now, unless something drastically changes, I think I am going to go with ProctorU again. It's not the greatest, but for me, I feel the most comfortable knowing that I can actually see that everybody themselves is taking the test. So I have that visual component, and that your browser is more or less locked out for the duration of that. So at least for biochem, at least I don't know about your end, but on my end, it went pretty smooth other than a couple cases the first time. So what I'll do for that is I'll put up instructions again, probably tomorrow. I don't know if I'll have time to do that today. And then what I'm thinking about doing is that if you go through the pre-checks of ProctorU and like your computer says all green check marks, you can send that to me. I'll put in like a Dropbox where you can send that image to me, and I'll give you extra credit on the test, just so I don't have to run into something like last semester where we were dealing with troubleshooting the day of. But like I said, I'll make an announcement about that on the actual Blackboard, so you have it's all in writing, so it'll all be there. So that's really all I have about the test itself. If you want to see what the test is going to be like, look on the previous tests on Blackboard. You can kind of see what I've been emphasizing, at least on that test. And if you've been here in the live sessions, or if you're watching YouTube, you should have a good indication of things I've said over and over and over again. Those are the things that will probably pop up in questions. So, if you've been paying attention and if you've been diligent in your studying, you should have no problem, hopefully. And if you're wondering, like my averages for Biochem 2 tests, they are actually better than my Biochem 1 tests. I usually get a slightly higher average, like I want to say in the low B's is the average, 80 to 82%. So yeah. Does anyone have any questions off the top of their head before we move on to actual material? All right then. So we were talking about glycogen, and so I've just backed up a couple of slides just to get us refocused, since it's been a while since Thursday. So we've done glucose to pyruvate. We've done glucose to ribose 5-phosphate. Now we're going to be looking at the whole glycogen, both synthesis, so that's what we're going to focus on today, and breakdown, which we talked a little bit on Thursday, and we're going to finish up right now. So we went over the structure of glycogen. It's just a bunch of glucoses that have branch points. We talked about the idea of glycogenolysis, where you add a phosphate to a glycogen molecule. So a glucose in glycogen, you add a phosphate to it. You make G1P. So you chop off that glucose. Then you can use a mutase to convert G1P to G6P, and that can go through glycolysis. And we looked at this scheme for regulating the enzyme that's phosphorylates a glucose molecule in glycogen. And I'll just briefly, again, touch on that because we will be looking at this scheme more today. So in the phosphorylase, so the word on the bottom is cut off. So this is phosphorylase beta, this is phosphorylase alpha, and it's the same enzyme, just two different forms. And generally, when you see alpha and beta forms of an enzyme, usually alpha is more active. Beta is usually less active. And active in terms of doing its chemistry. And along with alpha and beta, we have T and R forms, which we learned about back in biochemistry 1 when it came to hemoglobin. So we can switch between alpha and beta forms through phosphorylation. So if we add a phosphoryl group to our enzyme, we switch into the alpha form. And once you're in the alpha form, you favor the R state. So you favor this state in the alpha form. Glucose can inhibit this. And once we lose our phosphate, we go to the beta form. And the beta form favors the T state. And you can tell that by the thickness of the arrows. That is showing percentages or probabilities to be in each state. However, it is still possible to go to the R form in the beta state if you have AMP around. Remember, AMP is a molecule that says you're low in energy. So even though you're in the beta form, which is less active, you can still turn it on if the cell is really low energy. So that is where we left off on Thursday. So here are some questions, and just for the sake of a little bit of speed, I will just go and answer these couple of questions until we get to the next PowerPoint. So we have glycogen phosphorylase, and remember, this breaks down glycogen, which releases glucose. So the question is, why would glucose inhibit glycogen phosphorylase? As shown here in the alpha form, if you have glucose, you go from the R state to the T state. And to really answer this, you have to know what glycogen does. So glycogen is glucose storage. It's in your muscles, it's in your liver. This is your emergency supply of glucose to make ATP. And if you already have glucose around, you don't want to break your storage, right? You don't want to break down your emergency supply of glucose if the cell's already flushed with glucose. So that is why glucose can inhibit phosphorylase A, because the enzyme makes glucose. So why make glucose if you already have it available? And it's the same thing on the beta form. ATP or glucose 6-phosphate inhibits the beta form, because why turn into the active form, the R state, if if you already have ATP or glucose 6-phosphate, right? So both glucose, ATP, and glucose 6-phosphate, all of those are saying the cell's going to be high energy, so don't break down the storage. So questions about that idea, or does that idea make sense to people? All right. So the next question then is the glycogen phosphorylase reaction, that is taking glycogen plus a phospho group and making G1P. That's the glycogen phosphorylase reaction. And I guess you also have in your product also glycogen -1 residue. So this normally has a delta G of 3.1. Delta G not prime. 3.1, which means it's not spontaneous. So why in the cell is it spontaneous then? And this is another theme that I've said in just about every lecture, right? It's this idea of substrate channeling, this idea of pathways. Because what's happening is that this G1P, glucose 1-phosphate, via mutase becomes glucose 6-phosphate. And this glucose 6-phosphate goes through glycolysis. So if you're low in energy, right, and you need to run through glycolysis, that means all the G1P that you produce by doing this phosphorylase reaction is going to be used up immediately to make G6P. So by keeping the concentration of G1P low, you actually make the delta G inside the cell negative. And we've seen this with glycolysis as well, where we look at these delta G not prime numbers and they are non-spontaneous, but because you keep the product of the reactions low, you can continue to make it spontaneous. It's the idea of coupling again. Yep, exactly. So in Biochem 1, I joked that if you didn't see something, if you didn't know something, 10% of the time you say hydrogen bonds. Here, 10% of the time say coupling. Oh, I didn't put my previous exams on Blackboard yet? My bad. I should have those, so I will put those up. I'll make a note of that and do that after class. I thought I did that, but I guess I only did that for Biochem 1. I know I had previous exams. Yeah, I should. Because last semester I definitely have a previous exam for exam 1 and 2 before we went online. And the year before that, I also have exams because we were all in person. So I'll put those up. You saw the first one, but not the others. Yeah, I'll look at my folders. I'll put those all up. Thank you for letting me know. It's coupling. Coupling is always the answer. All right. So the last, not the last thing. So we talked about taking glucose from glycogen, adding a phosphate to it. However, phosphorylase does not work once you get near a branch point. So phosphorylase cannot work unless you are 5 away from a branch point. So what happens when you break down glycogen is you chop off glucoses until you get 4 away from a branch point. Then that enzyme stops working. And we said on Thursday, this is the rate limiting step of breaking down glycogen. Because to continue to break down glycogen, I have to move this branch. It can't be a branch anymore. It has to be in the main chain. And the way this happens is called glycogen debranching enzymes. What it does is that it transfers 3 of these sugars. So it transfers a trisaccharide. So everything in red gets broken off and it gets put onto a separate strand of glycogen. So it's no longer a branch. It's part of the main chain now, as shown here. Once you're in the main chain, phosphorylase can begin again, start making glucose 1-phosphate and stop chopping off the glucoses. Now, when we use the debranching enzyme, we will have 1 glucose that will remain on the branch. This can't be moved through debranching enzyme, but that we can chop that off. So what happens is we just have a simple hydrolysis reaction, and this leaves as glucose. So we don't have to phosphorylate it or anything. We just snip that last glucose off, the last one that was the branch. That becomes glucose now, so that can go through hexokinase and become glucose 6-phosphate, while everything else becomes glucose 1-phosphate. Like I said, this debranching method, that's why your muscles have a maximum capacity of only a few seconds, because what happens inside your muscles is that phosphorylase will work very fast until it hits the branch. Then you have to wait for the debranching enzyme to work. And that takes a long time in comparison to the phosphorylation reaction. So that is the debranching enzyme. So any questions about the information presented here or anything I've covered up to this point? All right. So once we have our G1P, and I'm going to rewrite the stuff that's cut off for you, we just need a simple reaction to make it a G6P. And my G's and my 6s have not gotten better. And this is through a phosphoglucomutase. And it's a super simple reaction. Not much to say here, really. Your enzyme that you use a serine. And your serine has a phosphate attached to it, as shown here, right? And what the enzyme does is that it'll make a glucose with 2 phosphates on it. It'll attach a phosphate to the 6th carbon. So you get glucose 1,6 bisphosphate. Remember, we talked about this before. When you see the word bisphosphate, that means the phosphate's on 2 different carbons. A diphosphate means it's attached at the same place. So you make a bisphosphate. Then to regenerate the enzyme, the serine takes the phosphate from the 1 position. The enzyme's ready to go. You have glucose 6-phosphate. You're ready to continue with glycolysis. So that's the 3 main steps of breaking down glycogen. Phosphorylase chops off glucose to make a glucose 1-phosphate. Debranching enzyme moves branches so you can continue with the phosphorylase. Mutase switches glucose 1-phosphate to glucose 6-phosphate, does it through a serine amino acid, which makes sense, right? Serine is one of the few amino acids that can be phosphorylated because it has an oxygen. So you need a side chain to end with an oxygen. Usually that's going to be a serine. Serine's usually what you look at to get phosphorylated. It can also be threonine, but it's mostly serine. Now, once you have this glucose 6-phosphate, you can go across glycolysis, or you can do the pentose phosphate pathway. We looked at both of those already, how glucose has different fates. If this happens in the liver, where most of your glycogen is stored, though, you can also have it travel through the bloodstream. In fact, this is one of the ways your body feeds itself. When you have not eaten in a long time, it will start to release glucose from your liver. But glucose 6-phosphate cannot leave the cell, which is generally a good thing. When you do glycolysis, the first thing you do is you add a phosphate to make glucose 6-phosphate. This is to prevent glucose from leaving. You trap it within the cell. But if you're trying to export glucose, this is now a problem. So the first step is to use a phosphatase and simply knock off that phosphate. So a phosphatase is the opposite of a kinase. A kinase adds phosphates generally. Phosphatases usually knock those off. Now, G6P, we talked about this before in passing, but glycolysis happens in the cytoplasm. But the glucose 6-phosphate enzyme, our G6P ACE enzyme, is in the ER. And normally this is very good. This is a good thing. Because imagine if this glucose 6 phosphatase was in the cytoplasm, every time you made G6P, this thing would just automatically reverse it. And that would be a big problem. You'd just be wasting ATP to do nothing. So to prevent that, we store the enzyme that breaks down glucose 6-phosphate in the ER. So if the liver wants to export glucose to the body, we need to get glucose 6-phosphate into the ER. And this is through a translocase. Translocase is simply a protein in the membrane of the ER that will move glucose 6-phosphate. Then you bring the glucose 6-phosphate into the ER, you hydrolyze it, then through another series of transports, you return to the cytosol, then it can be released into the cell. As you can see, there are a lot of moving parts. There's moving parts in breaking down glycogen. There's moving parts for taking glucose 6-phosphate in the ER and then moving it out of the ER. So if any one of these enzymes is deficient or it's mutated or doesn't work, that definitely causes disease. And the whole class of disease is called type 1 glycogen storage disease, where your glycogen storage really doesn't work the way it's intended. But any questions about the information there, what this slide is saying? Oops. All right, so I'm going to switch PowerPoints now. And if you notice the PowerPoint, whoopsies, wrong one. If you notice the PowerPoint I put up for today has the date January 30th or something like that. Yeah, I forgot to change the date on it. My bad. It should be for Feb 9th. And we're going to talk about the opposite thing now. So we just talked about and I'm going to redraw this. So this says there's a box down here that says glucose 6-phosphate. We just talked about going from glycogen to glucose 1-phosphate. We're going to talk about the opposite process now, how to go from glucose 1-phosphate to glycogen, how to store glucose. So that's the whole chart that's being shown there. Breaking down glycogen, we mentioned this in one of the questions. Overall, when everything's taken into account, it's negative. It's spontaneous. So the opposite process, going from G1P to glycogen, not spontaneous. So we need to put in an energy source. And this might be the first time you've seen this in your science careers. Our energy source is UTP, not ATP, but UTP. So here, uridine triphosphate is going to be the energy we use to go from G1P back to glycogen. The breakdown and creation of glycogen, they are separate pathways. They happen in separate parts of the cells, which makes a lot of sense because if this didn't happen in different parts of the cells, you'll just be doing this circle. And every time you do this circle, you waste a UTP doing nothing. And so you're going to run out of energy very fast. But we're going to look into this in depth today. So let's take a look at glycogen synthesis. So this might look intimidating, but let me walk you through it. Here's UDP glucose, and actually I should start all the way on the left. So remember, we need energy to make glycogen. So what we do is we take our glucose 1-phosphate that comes from the breakdown of glycogen and our UTP. You don't need to know what UTP really looks like. You just, for this, if you can explain to me like kind of the basis of what's happening, that's fine for me. But what we do is we combine UTP and G1P to make our UDP glucose. This UDP glucose is high energy version. And it's good that we create this high energy version because we're going to be sticking glucoses together to make our glycogen. And our energy is coming from these phosphates. So what we do is that we have our UDP glucose and we just add it onto our glycogen. You don't need to know the arrows for this mechanism or anything. Just know that G1P plus UTP equals UDP, and know that's what we use for glycogen. So glycogen synthesis is controlled a lot. There's a lot of allosteric control for glycogen synthesis. In fact, under normal conditions in your cell, the amount of ATP, ADP, and PI that's available will make sure that glycogen synthesis never runs, right? And you turn it off by phosphorylating it. However, if you have G6P in your cell, this can reverse phosphorylation. So that's why it says the dephosphorylated enzymes activated by G6P. So normally this enzyme, this synthesis enzyme's off, normally off. But G6P can turn it on. And just a quick question here for those of you who joined the live session. What's the logic of having G6P turn on glycogen synthase or synthetase? What's the logic of this? Why does that make sense? to signal UTP to make UDP glucose. That is what is turning on, right? Yeah, so that is the signal that is turned on. I guess my question is, why does it make sense that glucose 6-phosphate controls that? Why does it make sense that glucose 6-phosphate says, if I'm around, make glycogen, please? Anyone have any ideas on that? One of these big thinking, take a step back questions. Right. Well, let's think about this again. So to really understand these types of questions, we have to understand the systems we're working with and their roles inside the body, right? So this enzyme makes glycogen. That's all this enzyme does. Glycogen is glucose storage, right? It's how we store our glucose. The first step of glycolysis is we take glucose and we make it into glucose 6-phosphate, which turns on the enzyme to make glycogen. So the logic is, if you have a lot of glucose 6-phosphate around, that means you must have a lot of glucose around, and you're going to be making a lot of energy. So instead of spending all that glucose right now, take some of your glucose, since you must have a lot of it if you have a lot of G6P, and put it in your storage molecule, glycogen, until you are low energy again. So that is the logic. Don't spend all your money at once if you have a lot of cash. Put it in your savings account for when you retire and you're not making money anymore. It's basically the same idea, but with energy. This enzyme is called glycogen synthase. Sorry, I can't speak today. Yeah, it should just be called glycogen. Yeah, glycogen synthase. So yeah, the enzyme that controls glycogen synthesis is glycogen synthase. So any questions about that logic or that idea? And so throughout this lecture, I want you to keep that fundamental idea in the back of your head. Low energy states, high energy states. We want to break down glycogen if the cell is low energy. What tells us we're low energy, right? We want to store, or we want to make glycogen if we're a high energy. And what molecules tell us we're high energy? Because we're going to be looking at some flowcharts today that deal with all of that logic. Okay, so question 1 is just a kind of review. So I will answer this. I did mention it many times. A kinase adds phosphates generally. Phosphate. And a phosphorylase removes phosphates. And this is all general because people have named things wrong before, where it looks like a phosphorylase. Basically, they name the opposite process, where sometimes you will see a phosphorylase add a phosphate and a kinase remove a phosphate. But it's a pretty safe bet if you come across a new enzyme that you've never seen before and somebody's named it a kinase, that's going to add a phosphate. And conversely, if you run across a new enzyme and it's called phosphorylase, those usually remove phosphates. So that's something else we should keep in mind for today. Okay, so expanding on this idea of creating glycogen, the enzyme glycogen synthase cannot take two glucose together and make a new glycan chain. Glycogen synthase can only build on existing glycogen. It cannot make new glycogen, which can be a problem since glycogen synthase's whole job is to make glycogen. However, we have a clever solution to that in that we have a separate enzyme called glucogenin. And glucogenin, I'm showing right here in the image. This is glucogenin. And what glucogenin is, it's like the start point for glycogen. And what can happen is that you can attach a glucose that's already been turned into UDP glucose, which we just looked on the previous slide. You can take one UDP glucose and attach it to tyrosine 194 of glycogenin. Once you have that first glucose there, now glycogen synthase has something to work with. You already have your glycogen chain started, so glycogen synthase can now extend after you already have this base going. All right, so glycogenin really acts as a primer to get your glycogen started. And so inside your muscles, inside your liver, for every glycogen chain you have, in the center of that chain, you have a protein that just served as a starting base. Glycogenin is an enzyme. Why doesn't it end in ACE? That's a great question. And the reason is it's because humans name stuff and we don't always follow rules. That's the best explanation I can give. I'm guessing it's because historically, when people first studied glycogen, they noticed this protein in the middle and they didn't know it was an enzyme. They thought, and this is all a guess by me, but I'm guessing the first time someone studied this, they thought that all this protein is to build glycogen off. They didn't realize it's an enzyme that does it starts the whole process itself. So that's probably why it's not ACE, rather in. Why can't glycogen synthase build glycogen off of UDP glucose? That's a great question. And I am not 100% sure why it can't just stick two glucoses together. Go back on the previous page. And if you just look at the mechanism, so this is the mechanism for glycogen synthase. It just needs a glycogen residue that's so many glucose long. So by knowing, and I'm going to fast forward again. Sorry for jumping around like this. So we know that you must have at least 7 to 8 glucose molecules in your glycogen before glycogen synthase can work. What that tells me is if I was to draw the enzyme, so let's say this is glycogen synthase. What that tells me is that if this is the active site, like this little circle, this is the active site where we add new glucose molecules, so glycogen, that is, I would guess, buried within the protein. And this channel probably binds 7 to 8 glucose molecules. So the enzyme works by binding these 7 to 8 glucose already in glycogen, and once they're bound in the channel, then on the active site you can start adding glucose. So that's the reason I would think why, like on a molecular level, that happens. On a logical level, why this couldn't happen is because you really want a molecule that's like big and branched like this. If glycogen synthase had the ability to just stick 2 glucoses together, it's very likely you would just have like random small glycogens instead of this big thing of glycogen that the cell has. And I'm guessing just random small molecules of glycogen, they're not as efficient as this big molecule of glycogen. And so by having a system like this, I would imagine you can control how much glycogen's in the cell at any given time by controlling the amount of glycogenin, because this is what is starting our glycogen. That would be my guess. Yeah, you need like 8 glucose molecules to tyrosine 194. Yeah, that's right here. So once you have the first glucose there, then glycogenin will extend. Then glycogen synthase can start working. So this should be glycogenin. Whoops, and then. Can this be done to anything other than tyrosine 194? In theory, this could be done to anything with an OH group, but in reality, the enzyme's probably set up in such a way that the active site, tyrosine 194 is in the active site for this reaction to happen. So no. You can add a glucose to anything that adds an OH, but for this enzyme, it doesn't have the machinery, the amino acids around anything other than tyrosine 194. So short answer, no. All good questions though. Any other questions about our glycogen synthase though? All right. So glycogen synthase can only add alpha-1,4 linkages, which is great, except glycogen needs to be branched. So we need another enzyme to add our branches. And this is called amylose transglycosylase. In reality, it's called amylose 1416 transglycosylase. And what it's doing, and if you remember way back in biochemistry 1, we talked about how starch is made out of 2 different things. And one of those things was amylose. Amylose is linear glucose. So if I was going to translate this into more English, linear glucose, moving it from 1,4 linked to 1,6 linked, and that's what transglycosylase means, moving sugar. So this enzyme is named like linear glucose 1,4 to 1,6 move sugar enzyme. Transglycosylase. Not the same as the debranching enzyme. So remember the debranching enzyme, what the debranching enzyme would do is that it would take it and just make it like 1,6 just added to the long chain. Here, we're just adding it to another branch point. And this always does it in units of 7. So we always move 7 units of glucose at a time to make a branch. And the original chain must have at least 11 residues, right? So for the branch to move, has to have at least 11 glucoses. And we will move a 7 glucose unit to make a new branch. And branch points must be 4 glucose apart. And that's what's being shown in this image. So glucoses are at least 4 apart. Now, the beauty of this, once you have a branch, you can now start adding glucoses here, or here, or here, right? It's a really clever way to build up glycogen fast, because every time you branch, you just open up a new glucose that you can add, or yeah, new glucose to add onto your molecule, a new start point rather. And then when you're breaking it down, we already talked about this concept. When you break it down, you now have 3 points to break down glycogen from, when originally you only had 2. So this branching speeds up storage abilities, but more importantly, it speeds up how fast you break down glycogen, how fast you get glucose into the cell. Right. Any questions about that idea? All right. Now I know this seems like a lot of information, and it is, and it's very complex. But at least every time I go over this, it's always amazing to me like how intricate the cell is on like every single detail. And then once you think about how it works, it's just like, wow, that is one of the most logical systems you could come up with. It works beautifully, but it is hard to learn because there's a lot of moving parts. Right. So question 2, this is kind of a review question. So when you're going back to study, come back to question 2 with a fresh sheet and say, how do I build glycogen, starting from UDP glucose in the primer glycogenin, and what do I need? So I'll just go over this really quick again, right? What you do is you have glucose 1-phosphate plus UTP makes UDP glucose, right? So that's how we get our UDP glucose. It's from glucose 1-phosphate. Glycogenin, we have a tyrosine molecule, or tyrosine amino acid rather. This is where we are going to begin adding our glucose, our UDP glucose. So we add, glucose to this tyrosine, and we add 8 of those, right? Then, once we have those, glycogen synthase can start adding onto that. So it'll extend this. And to make branch points, that's where we have our amylose 1,4 to 1,6 trans glycolase. Again, remember, linear sugar, I'm taking it from a 1,4, making it to a 1,6. I'm moving sugar, transglycolase. That will create branch points. If you can answer number 2, and you have all that information down by the time it comes from the test, you have glycogen synthesis down. You should be good. And as long as you can tell me, like, what substrates do we need? What are the enzymes involved? Are there any cofactors? That's all I'm looking for when it comes to glycogen synthesis, that kind of basic information. So any questions about that? All right. So glycogen is buried in the glycogen particle. How is this different from most enzymes? Again, I'll just answer this really quickly. So most enzymes do multiple reactions. Glycogen does one reaction. And let me expound on that. What do I mean by multiple reactions? I mean that most enzymes will cycle, right? They'll do a reaction and release the product and be ready for the next reaction. So they take in another substrate, do a reaction, and they're good to go. Glycogen, or glycogenin rather, it doesn't do anything once it's done its purpose. It gets the glucose added to its tyrosine molecules, and then it's kind of like a skeleton almost, right? It just stays there as the glycogen body grows. So each glycogen molecule can only do one reaction before it's used up, which is a complete opposite of every enzyme we've ever talked about up to this point. Every other enzyme does thousands, millions of reactions before they're done. Glycogen does one reaction, then it stays in the middle of our glycogen molecule. So that's the big difference there. All right. Now comes, I think, some confusing stuff, because we've been talking about pathways. When we talk about glycolysis, that's a linear pathway. Sure, it can go reverse. And then we added some complexity onto it with the phosphate pentose pathway, and then some more complexity with glycogen. If it does one and stays in the middle, how does it do the other reaction? Well, so like it just stays in the middle, then adds like 7 glucose to itself, then it's done. It doesn't add any more glucose. Glycogen synthase is the one that comes in and starts making the rest of glycogen. But yeah, think of glycogenin as like a staging platform where it gets the reaction started and then just kind of stays around, trapped within our glycogen molecule. That makes sense? Yeah. So we're going to walk through these. Now I'm going to redraw what's cut off on the bottom. Phosphoprotein, phosphatase 1. And that's going over here. All right. We are now going to look at how glycogen phosphorylase is regulated inside the body, right? And remember, keep this in mind. Glycogen phosphorylase in the alpha form breaks down glycogen. So I have that written for you. In the beta form, it does not. And you've already seen this image before. It's where I had the beta and the alpha. So that's beta, alpha. I guess this actually, sorry, that should have been in green. My bad. This is the R form. And then you can be in the T form with beta, alpha, and alpha has a phosphor group on it. So this is breaking down glycogen. We're going to look at how we control the breakdown of glycogen inside the cell. So it's quite complicated, but it does make a lot of sense when you break it down. The protein that is important for our regulation is called PP1, phosphoprotein phosphatase 1. So I'm just going to call it PP1. So what PP1 does, and that's what's shown down here in this box, that's PP1. What PP1 does is that it removes the phosphate from glycogen phosphorylase A to make it into the beta form. So stops the breakdown of glycogen. That's mainly what it does. What it also does is that it goes up and affects this enzyme called phosphorylase kinase, right? So what does phosphorylase kinase do? This enzyme, phosphorylase kinase, phosphorylates glycogen phosphorylase, right? Confusing with all those words that sound exactly the same? You're not alone. So this enzyme turns on the breakdown of glycogen. So this turns on, so phosphorylase kinase turns on glycogen phosphorylase. PP1 turns off glycogen phosphorylase, and it also turns off the kinase. So PP1 is like a master switch saying, okay, the thing that actually breaks down glycogen, I'm going to turn you off. And the enzyme that turns you on, you being glycogen phosphorylase, I'm also going to turn you off as well. So I guess a good analogy is, like, let's say you're doing electrical work. What you're going to do is you're going to turn off, like, let's say I'm doing some wiring work to a fan, because I'm looking at a fan right now. What I would do is I would turn off the switch to the fan in my room. That would be like turning off glycogen phosphorylase A. I would then go to the breaker and turn off the electricity to that room to make sure it doesn't accidentally turn on. So that's like taking phosphorylase kinase out of the picture. I'm turning off the enzyme, and I'm also going to make sure you cannot turn on again. That's what PP1 does. All right. Let's look at PP1 inside the cell. PP1 inside the cell, so I'm looking at this image now, where I just drew the new arrow. That is attached to glycogen via another protein called the GM subunit. GM means glycogen muscle, because what the image we're looking at only works in the muscle. Right, so I'm going to focus on the top path. So if your muscle receives insulin, so gets the signal for insulin, what happens is that the GM subunit becomes phosphorylated at what we call site 1. Once it's become phosphorylated, PP1 is more active now. PP1 is turned on, which turns off the breakdown of glycogen. So breakdown of glycogen is down. while the creation of glycogen synthesis is up, which makes sense, right? Insulin means that you have a lot of glucose in your blood. So if you have glucose in your blood, don't break down your storage, instead store it up. While if you get another signal, epinephrine is your fight or flight, more or less, hormone, where it's saying you need to be ready to mobilize energy because you're going to be moving very fast very soon once epinephrine is released into your body. What that does, it turns on an enzyme called protein kinase A, PKA. If you've never heard of PKA before, and you continue down biology / biochemistry / molecular biology / health / being a doctor, yeah, you're going to hear about PKA a lot. PKA is used in a ton of processes to phosphorylate stuff. Here, what PKA does, if I'm just looking on the left picture and it's cut off here, so I'm going to redraw it, it adds another phosphate to site 2. If the GM subunit has 2 phosphates, PP1 is now inactive. PP1 gets released from the GM subunit. It's now inactive. What that does is, and I'm going to change my pen here. If PP1 becomes inactive, then this pathway is inactive, this pathway is inactive. Plus, what PKA also does, PKA, and I'm going to change the green here, PKA turns on phosphorylase kinase, which turns on glycogen phosphorylase alpha. So when you have epinephrine around, the breakdown of glycogen increases, while glycogen synthesis decreases, because epinephrine says, get ready to make energy, start breaking down that glucose. We're either going to be running away or throwing fists soon, or you're really nervous because you're going to be taking a test. So your body is going to produce a ton of ATP to make you even more nervous. All right. So I know these images, when you first look at them, can be really intimidating and really confusing to know what they say. So one way that really helps me is just going back a couple of slides and reminding myself, okay, what does glycogen phosphorylase do? And then relate that to what signals are working when. But just from that explanation, is there any questions people might have right off the bat? Okay, and I believe the next slide says what I just said. So if you need another explanation, it's on the next slide, and I'll just say what I said in fewer words. All right. You like graphs? Well, I have more graphs for you. All right. Now, we just looked at the graph in the bottom right. So this PPI. And what I'm showing you on the left is zooming out of the graph on the right. So they're actually the same image, just the one on the left is zoomed out. So let me draw like arrows showing you where everything belongs. So this is this, that is that, this is this. Then we have our phosphorylase over here. And this is PPI. And this is the inhibitor down here. So I'm going to get rid of these arrows in a second because they're super confusing. But even though the one on the left has more boxes, it's really what we just talked about. Okay, so this whole system of breaking down glycogen, it's all controlled by a simple molecule, or one of the major control points, cyclic AMP. So let's remember what cyclic AMP does. Once you remember that, it'll make things, I think, easier to understand. Cyclic AMP means low energy. Okay, when you're low energy, you activate pKa. pKa turns on. pKa's whole job is to go around and add phosphates to things. So if we follow the gold pathway, the gold pathway always means that CAMP is available. So that's what gold is. So gold, let me get gold. Gold is, you have CAMP. So what pKa does is that it will go and phosphorylate PP1 inhibitor, or yeah, PPI, more or less. So this is the inhibitor of PP1. All right. And what it will do is it will bind PP1 and make it inactivated. So when you have CAMP around, PP1 is inactivated. pKa goes and turns on phosphorylase kinase A that we just talked about. This will go and break in phosphorylase glycogen phosphorylase A. We are breaking down glycogen when this is turned on. It will also go and turn off glycogen synthase. So off. We are not making glycogen. And so that's all in the case if CAMP is around, cyclic AMP. The reverse case is shown in that purple. I guess I'm going to go with blue. So blue is no cyclic AMP around, which means high energy, which means pKa is turned off. When pKa is turned off, the inhibitor of PPI, or PP1, sorry, is also turned off because it doesn't get phosphorylated. So it is turned off. If the inhibitor of PP1 is turned off, that means PP1 is active. So that's the no cyclic AMP pathway. Once PP1 is activated, it will turn on glycogen synthase A. So we're making glycogen. It will turn off glycogen phosphorylase B. So don't break down glycogen because I already said turn it on. And it'll also turn off phosphorylase kinase B, which is another kinase that turns on the breakdown of glycogen. Right? So keep that in mind. The basic story here, no cyclic AMP, that means you're high energy. Make glycogen, don't break it down because we have plenty of energy. The reverse case, if there's cyclic AMP around, turn on pKa. Once pKa is on, that will turn on glycogen phosphorylase to break down glycogen. Cyclic AMP means we're low energy, break down glycogen. Also, turn off the synthase. Stop making glycogen. We're low energy, break it down. So that is what this image says. A ton of information presented in one figure. And if you've ever looked at like pathways in any papers, or if you were at the talk this morning for the new biology candidate at what, like 9:30, he showed pathways as well. We're starting to see how complex even the most simple processes in metabolism are. There are a lot of moving pieces, a lot of things that are all interconnected to each other. All right. Do people have questions about how this system of when to turn on glycogen synthase and when to turn on glycogen phosphorylase works? Or do you just need some time on your own to digest it before you can really see if you understand it or not, or really see if you have questions or not? Time to digest, yes. Well, if you are digesting, is pKa turned on or turned off? Ah, ha, ha. Ah, ha, ha. Hilarious metabolism joke. pKa would be turned off if you're digesting food because you don't have cyclic AMP because you have glucose in the blood. Yep. So what I would suggest is really give yourself time after listening to this. Come back to this image in, I don't know, a half a day, I would say. And look at that diagram without the notes you wrote or my notes and explain it to yourself. If you struggle on an explanation, check back with your notes. And if what you wrote no longer makes sense or you can't make logic of it, contact me and I'll be happy to walk through this again. Just to let you know, like every time I go to teach this slide, it usually takes me a solid like 5 minutes to like figure out, okay, what does this say again? Yeah, so it is complex. And I think we will just call it here. So we have a little bit to go until the end of test 1 material. So we'll cover that like half on Thursday. And I think I'll say this and I'll make an announcement about this. We will get up to that point on test 1, up to here on Thursday. Then instead of continuing on with like the rest of the stuff, I will save that for next week, Thursday. So we can have half the time to look over new stuff for test 1, half the time for a review session on test 1. What I would like, if you want to come to the review session, is bring questions, and I'll post this on Blackboard in case people have questions they want me to go over. And if there's no questions, usually what I've done in the past is I just kind of go over the slides again one by one until somebody stops me. So it'll be much better if you do have questions. Either you'll email them to me once you're studying, or we can go over them in class. Otherwise, yeah, so I think for Thursday, I will probably just move the folders back again because this stuff's really dense. So instead of a new reading guide, we'll just continue from there. I'll put a homework up covering what we covered. It is due on test day, but it is related to the test. So I would definitely suggest you do that homework before you take the test, just to make sure you got it down. Otherwise, continue your studying. Let me know if you have questions. I will put up the exam for test 1, or I'll put up the exams for the previous tests. That's what I meant for the previous years. And I'll see everybody later. Have a good one, everybody.